Welcome to my talk. My name is Chhaya Werner. I'm a postdoc at the University of Wyoming, and I'm excited to share with you a novel method for estimating parameters and modeling community ecology dynamics. We know that interactions between species, such as competition, have strong impacts on how species are distributed in the landscape, with consequences for community composition and diversity, demography of populations and species, and even the functioning of ecosystems. And yet, investigating how those interactions play out in real communities can be challenging. For example, in this Australian gumjam woodland understory, a highly diverse community is also dependent on abiotic factors, including different levels of shading from the overstory canopy and a gradient in nutrient loading from agricultural runoff. In addition to their direct effects, these abiotic factors can shape the competitive interactions of neighboring species. We might want to know, for example, how patterns of population growth differ between a native forb, Waitsia acuminata, and an exotic forb, Arctotecta calendula, and mechanistically whether these differences are driven by differences in the species' intrinsic growth rates, or interactions with their neighbors, or both. And we would want to know how these differences vary depending on the abiotic context. This kind of information would be useful for understanding the community dynamics and for managing these populations. The existing challenge with these kinds of questions is that doing this analysis requires a large number of parameters, especially in diverse communities. For example, the native forb Waitsia is potentially competing with 45 co-occurring neighbors. Allowing species-specific competitive terms to vary by even one environmental gradient would result in 90 parameters for competition, a prohibitively high number. Because of this, we often make a priori choices to only consider a small subset of species and ignore the rest, or to lump species into functional groups by life history traits or origin. Ideally, we'd like to be able to model population dynamics in diverse communities, considering direct abiotic effects on growth rates and competitive effects from many neighboring species that can themselves depend on the abiotic context. And we'd like to do so in a way that maintains a manageable number of parameters and produces easily interpretable results. I'm excited to share a method with you that aims to do just that. I'll explain our method, which pairs a classical ecological model of population growth with a Bayesian method for selection of variables called sparse modeling. This sparse modeling framework is used in physics, genetics, and economics with linear models, and we've extended it to apply to community ecology models, which are usually nonlinear. I'll show you how we tested our method with simulated data and walk through an example application to a real empirical dataset. For this sparse modeling approach, we take all of our parameters for competitive interactions on our focal species, and we start by combining them together into generic terms, with all the interacting species lumped together. Here the large dots represent one generic term for the abiotic independent interactions, and another term defining the slope of how these competitive interactions vary with an environmental factor, such as shade or nutrients. We then let the data inform which species' competitive interactions differ from that generic term in their average competitive strength or in how their competition varies with the environment, and we model these interactions with species-specific terms. So how do we implement this mathematically? We start with an ecological model, such as this Beverton-Holt model of fecundity. In this equation, subscript E is the environmental condition, I is our focal species, J is each interacting species, and T is the time step. This equation describes fecundity as a function of a species' intrinsic growth rate, that is, with no competitive effects, its intraspecific competition, and the interspecific competition from its neighbors. And although I'm using the Beverton-Holt model here, the sparse modeling framework I'll be discussing can be used with a variety of ecological functions. To build our sparse model, we start somewhat counterintuitively by splitting some of our parameters into multiple terms. We split the intraspecific competition term into an environmentally independent intercept and a slope term describing how intraspecific competition varies with environment. We do a similar thing for the interspecific competition term, but here we have a generic term averaging across all interacting species, one for the intercept and one for the environment by competition slope, and deviation terms describing how those competitive effects differ by individual species. So how do we turn this into ultimately fewer parameters? We use prior distributions for our parameters that determine how likely they are to be included in our final model. These priors are normally distributed around 0, with a standard deviation defined by two values, tau and beta, that determine how tightly centered on 0 this distribution is. Tau is our global shrinkage parameter, which affects all species in our model. The smaller tau is, the more these species-specific competition parameters are pushed towards 0. Beta is a species-specific parameter that allows individual species to escape from this general push towards 0. When beta is large, the prior distribution is wide, which allows the parameter to become a non-zero number informed by the data. This prior is named a regularized horseshoe prior because if you calculate the probability a species-specific deviation is included, it forms a horseshoe shape. This approach allows us to use the data to inform which species-specific parameters we should keep and which can be lumped together in a generic term. When we tested this approach with simulated data, we were able to successfully recapture population growth rates on out-of-sample data, here with a root mean square error of about 0.3. What's more, we were able to fairly accurately estimate the underlying parameters. Looking here at the deviations between the true parameter and our model estimates, from the top to the bottom, we have the intrinsic growth rate lambda, intraspecific competition, a species-specific competition terms, and then the generic competition term, each with both an intercept and an environmentally dependent slope. We tried this with different input sample sizes, and we were able to capture population growth dynamics with just 10 community data points and 10 no competition individual samples. To repeat that, because it's pretty cool, with just 20 data points, we were able to pretty accurately estimate population growth rates in a variable, diverse community across an environmental gradient. With more samples, the model identified more species-specific parameters and further improved in growth estimate accuracy. Compared to the fully 30 competition parameters that would be necessary to model this 15-species community in the traditional way, our sparse models had only 4, 6, or 12 competition parameters, depending on how much input data was used. Now I'd like to show you an example of how this approach can be applied to an empirical dataset, returning to the diverse and beautiful York gum jam woodlands. This data I'll be using was collected by Claire Wainwright, Margie Mayfield, and Kath Bowler. One question we had with this data was how competitive interactions varied for different focal species and across environmental conditions. Here for the native forb Waitsia acuminata, with the phosphorus gradient on the x-axis and competition parameters on the y-axis. The generic interspecific parameter is shown here in black, with the intraspecific competition in blue and 2 non-generic competitors in orange. One thing to note is that intraspecific competition is generally above the generic interspecific competition across the environmental gradient. Something else interesting is that we identified both a stronger than average non-generic competitor and one with overall lower competitive effects. In contrast, here's the non-native Arctotheca calendula. For this species, the intraspecific competition, in blue, is generally lower than the interspecific competition, and we saw no non-generic competitors for this non-native species. In addition to the results shown here, we were able to quantify how intrinsic growth rates, lambdas, for these two species differed across the environmental gradients of phosphorus and shade, and based on this information predict which plots they'd be most likely to persist in or invade into. Using this model, we discovered that the native forb has higher intraspecific competition than interspecific competition, a key component of coexistence in communities. In contrast, intraspecific competition for the exotic forb was relatively low. I'd like to point out, that small blue dot should be a little more to the right to represent intraspecific competition for the exotic forb on itself. And the native forb had specific interactions with some of its neighbors, perhaps due to a shared evolutionary history, while the exotic forb did not. So to conclude, we're really excited to share with you this novel method in community ecology. We think that it is particularly useful in high diversity communities or in systems where data is particularly limited for one way or another. It's flexible in terms of the underlying ecological model used in a variety of ways that I'd be happy to discuss. And it's really great because it allows us to split out direct effects of environmental variation on intrinsic growth rates versus indirect effects of environmental variation through competition. The results that it gives are hypothesis generating as well as being easily to interpret. You can see how these outcomes from analyses of observational data sets could lead to both understanding of how community ecology works as well as setting up new experiments to test mechanisms. We think that it presents opportunities for bespoke modeling for management questions and applications, and we're excited to extend it to different types of data, including time series. Thank you for listening to my co-authors, members of the Estuareation Working Group, Chhaya Werner, who collected the data, our funders, and especially to the ESTIV Center, which coordinated and funded the working group workshop.